Thanks a lot, Sumantra and the organizing team for giving me this opportunity. This is a very special thing, especially because this is the first offline talk I'm giving after COVID. So, good evening. My name is Kishore. I'm from Dr. Mohit Kumar Jolly's lab at IISc. We have been working on uncovering design principles of complex regulator networks that are underlying cellular decision, cellular fate decision systems. Right? The system that I'll be talking to you about is EMP, which I'll get to in a minute, where we are looking at the regulator networks underlying EMP, trying to understand how multiple phenotypes come up in the process of EMP and how these phenotypes are stabilized. So epithelial mesenchymal plasticity, EMP, is a process of cells which undergo EMP reversibly switching between three broad category of phenotypes. Okay, so these phenotypes look something like this. We have the epithelial phenotype, which is more adherent. We have the mesenchymal phenotype, which is more invasive, and then we have the hybrid phenotypes, which are more prominently seen in the context of metastasis than the development where EMP originally comes from, where the hybrid phenotypes have the property of both epithelial and mesenchymal phenotype. Right now, there is a lot of complex and interesting biochemistry that goes on with each of these phenotypes. But what we are particularly interested in is how this switching happens reversibly between these phenotypes. And there have been biochemical and biomechanical signals that have been shown to induce switching in one direction or the other. Okay, so and such multistability, especially in the context of metastasis, can enhance survival and indirectly has also been shown to enhance survival of metastatic cells. Right. So the first question is, how do we have actually multiple phenotypes that underlying EMP? And the second question is, what do these transition rates look like? How stable are one of these phenotypes? Now, there have been again multiple experiments to sort of construct the EMP phenotypic landscape in a very colloquial sense. So one such very famous experiment done in vivo takes a heterogeneous cell population which has multiple cells of various different phenotypes. I've broadly colored them as epithelial, mesenchymal, hybrid, and they isolate them into one homogeneous population and then let the homogeneous population repopulate within within a mice and see how much of the heterogeneity that was existing before has been recapitulated. Right? And based on this, they were able to construct this semi-quantitative landscape. Here you see the transition rates are broadly represented by the thickness of these arrows. Okay, and there I would like to draw your attention to two kinds of phenotypes here. I know we have three phenotypes: epithelial, mesenchymal, and then hybrid. But the epithelial and mesenchymal phenotypes have a common characteristic in that they're both highly stable. So the transition rates of the cells in epithelial or mesenchymal phenotypes are lesser as compared to the transition rates for hybrid phenotypes, which are at the middle. Okay. So the question is, can regulator networks actually explain these stability patterns? And to answer this question, we collected a bunch of regulator networks underlying EMP, which were constructed with various different methods and in different contexts of cancer as well as in vitro in vivo data. And we took these networks, asked if the dynamics of these EMP networks, first of all, can they recapitulate what was seen experimentally, and if we can find aspects of the network topology that can actually explain the stability dynamics. So that's the hypothesis, that underlying regulator networks can explain multistability and switching dynamics. So I'll first tell you what we found in the network and then show you the stability aspects of them. So these were the networks that we looked at, and at the first look they look very complex. We tried to look at the interaction matrices, which each cell shows one direct interaction within the network, and the interaction matrix is often quite sparse in most of the biological networks that we've been looking at so far. So instead, what we did is we converted the interaction matrix to what is known as an influence matrix. So instead of taking single edge effect of node A on node B, what we are considering is we are considering effect mediated through multiple nodes, right? So each path can be considered starting from A to B, constituted of more than one node, is considered to construct these influence matrices. And this is a formula for calculating the influence matrix. And once we calculate the influence matrix, we did a simple clustering. And what we find is that there are clearly two teams of nodes in the network. And these teams we could identify the biochemistry of these teams, and clearly classify them as mesenchymal team nodes, which are highlighted in red, and epithelial nodes, which are highlighted in green, blue, sorry. And so this network, this complex network now can be simplified into this simple case where you have two teams each inhibiting each other, shown by the negative influence of blue cells, and activating themselves, shown by the positive influence of red cells. So now, given this, we wanted to ask if this network can first of all replicate the dynamic scene experiments. So we simulated these networks considering only the network topology. So obviously we went to Boolean simulations, and even in Boolean simulations we adapted a threshold-based system where each link here is given only a weight of +1 or -1 depending upon whether it's an activation or inhibition, and each node only has active or inactive, only two levels. Right. And a node is activated if that's not here. Yeah, a node is activated if the net activating links that are active for the node are more than the net inhibiting links active. So we simulated these networks for 100,000 random initial conditions, and up to a steady state is reached. And these steady states we plotted in this manner, where each black box in the matrix tells you the activity of that level, whether it's active or not. So black box is active, white box are inactive. So again, as you can clearly see, we can recapitulate the three phenotypes or three groups of phenotypes that we see experimentally. And these three groups of phenotypes are again identified based on the biochemistry of the nodes involved. So in epithelial phenotype, for example, we have the epithelial nodes active. In the mesenchymal phenotype, we have the mesenchymal nodes active. And hybrid has both epithelial and mesenchymal nodes active. Right. And if you remember, the influence matrix was also very neatly segregated based on the epithelial and mesenchymal nodes. Right. So there is already a correlation between the influence matrix of the network and the state configuration. So we looked at the frequency of these states or phenotypes. The frequency is how many of these random initial conditions converge to one or the other of these phenotypes. And turns out 95% of the initial conditions converge to epithelial or mesenchymal phenotypes. So that again already tells you that the epithelial and mesenchymal phenotypes are much more stable as compared to the hybrid phenotypes. We didn't stop there. Okay, before I get to that, yeah. So we looked at the correlation matrix. This is something that we generally see when we are looking at high throughput data, right? Correlation of the expression levels between all the different genes that we are interested in. And what we see is that the correlation matrix that we obtained from Boolean looks very similar to the influence matrix that we have. Okay, and so we wanted to see if this is something specific to the networks that we're looking at. So what we did is we took these network topologies and shuffled edges on random so that the topological features are more or less lost. And we calculated the difference between influence matrix and the newly generated correlation matrix, and the formula is given here. And what we see is that on the y-axis is the percent difference between the correlation influence. On the x-axis is the strength of the team that we get. So as you can see, the strength of the team varies quite a bit. The wild type strength, the strength of the original network is somewhere around 0.7, and the random networks have a strength of anywhere between 0.8 to 0. Right? Even though the strength varies so much, the difference between influence matrix and the correlation matrix is only up to 2% here, and across all the networks that is simulated, it's only up to 3%. Okay. So that's something interesting, and we're not entirely sure why this is happening, and we're looking into that. But the difference between influence matrix and correlation matrix is also the same when we obtain the correlation matrix using an ODE based method of simulation. So one thing is for sure is that the influence matrix is able to explain or show the most dominant phenotypes of a network. Now we want to look at the static and dynamic stability. And the static stability is defined as how well is the state supported by a network, in other words, frustration. So if the network topology agrees with the state, then there is no frustration at all. If some of the edges don't agree with the state configuration, then there is frustration. So we calculate the fraction of edges that don't agree with the state. And similarly, we did an experiment, a digital experiment, which is replicative of the experimental experiment that we've seen in the beginning, where we take a homogeneous population of cells, digital cells, we apply random noise and see how much of heterogeneity is recapitulated. Right? And this we call coherence, is the fraction of cells that retain the original phenotype of one perturbation. So these are two stability metrics along with the steady state frequency that we saw. And when we plot the steady states that we obtain on these stability landscape or stability metrics, we clearly see two groups of phenotypes, hybrid and terminal phenotypes. The terminal phenotypes consist of epithelial and mesenchymal phenotypes. Yeah. So clearly there is the semiconductor landscape can be obtained just based on the network topology. Now, the question is, do teams actually have anything to do with it? So again, we went back to random networks. And as I've shown before, the random networks have generally low team strength. And we expected that the terminal phenotype stability, which was higher for wild-type networks, should be lower for random networks. And the terminal phenotype stability can be measured by these two metrics, maximum coherence and minimum frustration. So the higher the coherence, higher the stability, and therefore they are terminal phenotypes. And as you can see, as the team strength increases, you have a high coherence. So the terminal phenotypes are very stable at high team strengths, but at low team strengths, there seems to be some sort of a distribution, which means that if you have strong teams, then you would see strongly stable terminal phenotypes. Now, we know we've to some extent explained the stability characteristics. We wanted to see if we can explain the phenotypic transitions also. So this is another experiment done in vitro, where a group of again homogeneous epithelial cells were exposed to TGF-beta, which is a EMT inducing factor. And what they see is at low levels of TGF-beta, most of the cells remain in epithelial phenotype. At high levels of TGF-beta, most of the cells go to mesenchymal phenotype. And at medium levels of TGF-beta, some cells are remain in epithelial, some cells are in mesenchymal. There is a bimodal distribution. So the question is, can these trends be explained by teams? So what we did is we did a similar experiment, and we subjected the homogeneous population to low, medium, and high perturbation, the perturbation being the fraction of nodes whose expression level is switched. And what we get is at low perturbation, most of the phenotype is what we start from, which is epithelial. At high perturbation, most of the cells are now switched to a mesenchymal phenotype. And at medium perturbation, we see a bimodal distribution. Now, what we did is now we did the same experiment on random networks, where 30 seconds. Yeah. So in random networks, what happens is that the insensitivity to low levels of perturbation for epithelial phenotype vanishes. Okay, and we fit these transition curves to sigmoidal function, calculate the cooperativity, and see that the cooperativity for terminal phenotypes decreases as the team strength decreases, or increases as the team strength increases. But for the same for hybrid phenotypes is not really affected by the team strength. So in addition to that, we also found that networks which have strong teams provide structural robustness to edge deletions, as well as having random weights attached to the edges. So in summary, EMP networks could be designed to have teams, as we see in the random networks having lower team strength. And strong teams lead to stable terminal phenotypes. And these terminal phenotypes have unique dynamic characteristics which have not clearly shown here. But this is something interesting for from a biology perspective. And teams do provide robustness against biochemical noise and perturbations. I just end with this slide where we have metastasis. That is a hugely complex process. EMT is just one aspect of it, one aspect of how metastatic cells survive. But there are multiple other aspects, and there are multiple other complex regulatory networks that underlie all of these different aspects, such as cancer stimulus, chemoresistance, etc. Now, what are the different patterns in complex networks that we can find? With that, I'd like to thank once again the organizers, as well as our team, Varun, Aditi, Archana, who helped me with this work. Thank you very much.
So we have time for one question.
Hey, nice talk. One thing I didn't quite understand, these teams that you're talking about. So in biological networks, people have seen for a very long time that there exist these modules and regulomes and so on, right? So conceptually, how are your teams different from these modules that people find in all biological networks?
So there is a lot of similarity. The merit of this work is, I would say, not really in identifying that there are teams, because we have known that there are teams, although no one has really pointed out that such teams would lead to this robustness. So that's one thing. And the second thing is establishing the pipeline to sort of identify whether teams are there in the network or not, and if teams are there, is there a causal connection between teams and stability of the landscape. So that's the main aspect I'd like to focus on. So not really, because the definition of modules is something that's not established. There are multiple ways in which a module can be defined. So I would say that there are common points between modules and teams. But one striking difference is that nodes of a same team do not need to be connected by direct links, whereas modules in general, we see that there are direct links connected. So that makes it sort of a higher order metric of cohesiveness in the networks as compared to the modules that we have seen so far.
Once again, thank Kishore.